Giulio Superti-Furga from CeMM Vienna speaks about biological, chemical and societal engineering for all-round innovation.
A very warm welcome. Good morning in the Österreichische Gewerbeverein, the Austrian Trade Association. I'm very happy that you're here. One word to the Austrian Trade Association was actually planned together with the Vienna University of Technology, which was founded then in 1814-15, but it was not allowed to be founded itself, so we had to wait 25 years until 1839 until this association was founded, political reasons. Nevertheless, there's a strong connection to technology, and this is the entrepreneurial part of using technology, using science. And this is exactly where we came together today. These Exner Lectures are honoring the professors Giulio Superti-Furga of CeMM in Vienna and Professor Ferdi Schütt of the MPI in Mühlbach an der Ruhr in Germany. The lectures themselves are to bring you and us together. It's a connection, it's a networking event. The laureates, the Austrian scientists, we want to be a platform of exchange and network, and we have a lot of room for exactly this. Besides the lectures, you have the program, found the program at the desk at the beginning. I will announce it as well. We are exceptionally happy to have a poster session, which is a competition actually, Milestones for a Future Worth Living. And believe it or not, we had too many proposals. So we had to select, which was painful, but that means if you are here with a poster, it's a success by itself. You already won. Little housekeeping, we will have 2 sessions, one morning session, one afternoon sessions. We have a lunch break in the middle, and after the second session, we have a coffee break, and then we run into the second part of the poster session. The poster session will be during the lunch break, and after the coffee break in the afternoon. For the poster presenters, please be in the proximity of your poster to answer questions, maybe present a little. Your job will be to contribute, to network, and to enjoy. The medal was, the Wilhelm Exner medal was founded 1921, honoring Wilhelm Exner himself. He was born 1840, so he was 80 by that time, and he led his life by connecting people, bringing them together, enabling technology, enabling networking, and taking care of a very strong leg in taking care of education. Urania was founded by him. The TGM was founded by him. The place where the Boku is today was made possible by him. And therefore, this medal is to honor those who have promoted science, in particular for achievements and developments that are suitable for direct applicability and implementation for the benefit of the economic development and for the improvement of the living conditions of the population. Now, the translation to the work of the laureates is very easy. You heard why they received the medal tomorrow, actually. And I now open the session, Professor Giulio Superti-Furga, Research Center for Molecular Medicine in the Austrian Academy of Sciences in Vienna. We will hear talks with cutting-edge medical and translational research from scientists from CeMM, MOLIT, Molecular and IT in Heilbronn, and the MedUni Vienna. I will not preempt the laudators tomorrow, and I ask the laureate to the stage. The talk is titled Biological, Chemical and Societal Engineering for All-Round Innovation.
thank you very much. It's impossible to describe the honor that one feels about being among this amazing list of laureates. So I thank Stefan Blahut. I thank the Wilhelm Exner Medal Foundation for choosing me. There must be a mistake somewhere. And the fact that today there is a talk and tomorrow is a medal is that if you don't give a good talk, you don't get the medal. So there is still a chance to escape. I would like to start by thanking a lot of people, certainly thanking my team over the years, the teams that started the companies over the years, my mentors, without whom nothing would have been possible. Each one of us, and you will see in your own career and life, each one of us constantly encounters people that are actually helping. Sometimes you don't even know that they are helping you, but they really are helping you. And without that, it would be very difficult to traverse all the different stages and obstacles. And so, on top of thanking then also my family, I would like to thank you for coming. I think that, and certainly I would like to congratulate Ferdi Schütt as the other laureate today, because young people doing research are really what it's all about. And we know that you will find fantastic solutions for present and future problems, and we also know that you will carry the torch into the next generation, and so on and so forth. So what I would like to tell you today is a little bit about the research we're doing now in the last, let's say, years, but also at the end tell you a little bit about how I see innovation in the area of, let's say, biochemistry, life sciences, and beyond. So I would like to start by reminding you that we don't quite know what the origin of life was, but for sure there was the ability of molecules, organic molecules, to somehow self-assemble into some sort of polymers. And it's the ability to separate this assembly from the environment that allowed the system, if you want, the early biological system, to create an own chemical environment. And separating sort of the system from the environment is fundamental, not only to maintain the genetic identity that is associated maybe with these replicating polymers, but also because you are therefore maintaining a different concentration of molecules. And so the order that you're able to create in the biochemical system clearly sticks out compared to the disorder that you typically have in your environment. So we need two things. We need, for sure, the membrane that is separating the endogenous, the inside of the system, and you need molecules that are able to provide this genetic identity so that the biological system replicates. I tell you this, and this is from an artist called Urs Wehrli, who likes to put things in disorder and order, and I find this soup reminding me of the Ursuppe, that you know may be associated with origin of life, of how a certain order requires energy and is intrinsically associated with a lot of information. And then you develop this cell, right? This prototypic cell, whatever cell, all of your beautiful poster more or less have something to do, many of them have something to do with biology or biomedicine. They all require some sort of living system, whether it's an insect cell, whether it's a bacterium, or whether it's a human cell like depicted here. You have a constant turnover of chemical matter. You need to have access to the chemical matter with sort of access to nutrients, but you also need to be able to get rid of the waste that you don't want, because you are preserving a homeostatic chemical environment that is conducive for the biochemical reactions that are important. And so how is it all governed? And I like to use this print of a medieval print or a Renaissance print of ancient Rome. Ancient Rome had apparently 16 gates, and these gates were governing the access to the city. As you know, you want to be able to keep certain things outside, like, I don't know, barbarians or other or threatening Huns. And at the same time, you would like to bring in what you need, which is nice visitors, and of course, olive oil or wheat or white or red wine or whatever you need. And so it goes without saying that these gates need to be regulated. These gates are not sort of, they're open at all times, because if they weren't open at all times, you hardly would need them. And so this is reminding us about the biological system. How is it that the biological system regulates access to chemical matter, and how does it come about? And of course, you will hear more from my colleague Harald Zitte later about transporters, but I think it is this environment, there is a mistake in the spelling, it's this relationship with the environment that we like to study and we would like to understand. In a nutshell, there are many different ways how you can get small molecules or semi-small molecules through membranes. There may be a little bit of diffusion depending on the polarity of the molecule and depending on the time and the concentration, but most studies now say that the diffusion is not that important or it is not that determining, meaning that it occurs, but most of it occurs actually through transport of molecules through proteins. And the proteins, there are channels, and channels are those proteins in the membrane that allow the perfect connection between the outside and the inside, and therefore need to be regulated, because else you would lose a lot of the sort of potential of the membrane if you would do it, so you need to regulate it. But in contrast, the majority of transporters have an alternate access system. So they either are open to the outside or they're open to the inside. I think this is very important for my talk, because the three types of transporters that are shown here are all regulating access in an alternative way. And I'm not going to go into much detail, except to say that we are convinced that this regulation of these transporters that are sort of accessing or not accessing nutrients, and therefore the potential to make metabolism, I know it goes back and forth, I don't know whether it disturbs you, for me it's fine. The access to the outside of metabolites is fundamental to maintain the proper metabolism, the metabolism that is associated with you, with your cells, with your properties. At the same time, metabolism is associated with cellular states. Undoubtedly, a liver cell will have a different metabolism than a muscle cell, and so on and so forth. That in turn allows us the equation to say, if this is true, if transporters have something to do with metabolism, metabolism has something to do with the cellular state and identity, then probably also transporters have something to do with the cell identity. And therefore, if you were to change transporters and therefore access to chemical matter, you would probably modulate the ability of a cell to do something, to be itself. And if it's a cell that has a disease state, you can maybe correct it. If it's a cell that has lost its identity, maybe you can restore it. Or if it's a cell that has an identity, maybe it gets confused if you are confounding it with access to metabolites and nutrients that it usually would not have access. It turns out that most transporters in the human genome, the genes, are not characterized. We just don't know what they transport. Some of them are ultra well characterized, and you will hear more about this later, but not more. And so we think that there are maybe as much as 1,500 human genes encoding transporters, but there is a group that is particularly large. It's called the solute carrier. They are a superfamily. They have a different, we'll see that in a second, different evolutionary origin. They transport a variety of substrates or cargoes, and we think they reflect the metabolic state of the cell, because a cell will only require certain things and therefore open the doors, the gates to certain things. And we know by genetics that are implicated in a series of diseases. We'll hear more about that as well. What is also interesting is not only they are drug targets, as for example the targets of serotonin uptake inhibitors. I mean, they are very important for anxieties, but also the targets of liphoxins in glucose reuptake. So there are many important, very important blockbuster targets. But they are also transporting drugs themselves. This is a unique case in pharmacology where you have the same class of protein responsible both for pharmacokinetic and pharmacodynamic. And the Professor Zito will correct me, but I think it's quite a unique situation. So you have a situation where also the drug disposition is depending on transporters, because drugs also require this to enter, and we show this. And it's difficult to say exactly how many of these we understand of the SLC, but by my calculation, the majority of them are what you can call orphan, meaning that we don't quite know what their function is, we don't even know what they transport. And when we know a little bit about what they transport, because they're similar to something that is characterized, we really don't know about their function. Okay, this is a so-called uprooted tree. It means that at the center, it doesn't imply a common phylogenetic origin. It just implies that they are related because they actually come in different folds. This is all these solid carriers, and here are pictured the different in colors. For example, the light blues will be the one that transport amino acids, the other one, and so on, sugars, and so on and so forth, so that you can decorate the known SLC tree with the suspected cargos. And you see some parts are more homogeneous, some are less homogeneous, and there are some branches of these phylogenetic subtrees, some clades that are clearly heterogeneous also in terms of, and we think there are something like 23 different folds with a few folds that I will show you in the next slide that actually tells you about sort of the kind of transporter mechanism that exists. One very important mechanism is called the rocker switch of the MFS fold. This is a typical scissor situation where you are accepting the cargo outside of the membrane, and then there is an intramolecular restructuring that allows the cargo to go through. But there are also two different important mechanisms, and also the other folds usually uses. One is a gated pore, which is a lid, which is a kind of access to a way of transferring the molecule through a change in the conformation. And last but not least, there is several folds use the so-called elevator mechanism by which part of the molecule binds the cargo, moves the cargo across the membrane, and then releases it. So you see that there are all ways by which you can have this alternate system, and enough to say that not all of these are well drugged, and not all of these are well understood. But we are having a revolution in transport biology because the structures of the human proteins are now being elucidated at an incredible fast rate, mostly through cryo-EM. Okay, so how do you study these transporters? I mean, there are many ways. One is you can try to inactivate the gene for a transporter using human cells, one at a time, and then you see if I mutate this gene and it's not there anymore, of course you have to do both alleles, what happens? What you can also do, you can overexpress one. You can say over the background, I have more function by upregulating its function. You can also do it pairwise. Of course, you can do yet more. And you can also do it, let's say, in a random fashion where you select for a phenotype based by a random inactivation of all the transporters one by one in a pool. And so these are all methods that we have used, and I will just give you sort of an overview of a few situations that we have encountered. For example, Enrico Girardi showed that a lot of drugs that were killing this particular cell line that we were using were actually requiring transport for action. Not always were we able to show that it is actually for transport. So there could be some effects that are indirect, but most of the drugs require that. And that needs to, in effect, changes the ways that some of the pharmacology textbooks imply that due to some physicochemical properties of the agent will not require transport and will be permeable to cell. We believe that that is really not quite the case. We have done a lot of metabolomics and ionomics based on overexpression of individual transporters compared to the loss of function. We have also looked at protein interactions, and some of them were very nice. For example, we found an interaction that is regulating the activity of mTOR, a central metabolic regulator, and I will tell you a little bit more about a few others later. More or less, whatever we were looking for a particular cellular function, and I challenge the students to come with me with a cellular function that does not require a transporter somewhere, somehow, we found transporters. But one that I was really keen to do an experiment was if our theory is true, that there is a relationship between the cellular identity and transporters, then perhaps it should be possible to find a situation where a transporter that is localized at the cellular membrane. I forgot to tell you that not all transporters are located at the cellular plasma surface. Some of them are also located within organelles. If we would find a case where there is a surface protein that affects something as intimate, as central for decision as the state of chromatin and therefore the transcription of genes, then we would be able to prove that there is indeed this crosstalk between the environment availability and the inner making of the decision process of a cell. And this is something that Kai-Chun Lee, together with colleagues, were able to show, that sort of adenosine and adenine, sort of the ability to take up purine, was essential for a protein called BRD4 that is modeling chromatin using a genetic trick that Stefan Kubicek at CeMM had developed. And I'm not going to take you through this. Sufficient to say that for me, that was a nice proof of concept that indeed something that is happening outside the cells, exogenously added purines, would alter something as fundamental as a chromatin state in the center of the nucleus, and therefore gene transcription. Then if you start reading literature and understanding that a lot of what is happening to gene transcription, and Georg Winter is like to tell you more about gene transcription, and probably also Martin Jechniger, you will have a situation where the methylation of DNA, the methylation of histones, the acetylation of histones, and so on and so forth, many of the modifications that we know are associated with differential regulation of gene expression actually require an interaction with metabolism, obviously, because you need the methyl group, you need the acetyl group, and so on and so forth. And hence, transporters are associated with the molecules that are influencing the intermediate metabolism that then is linked to these processes. So I think this is a very interesting area of research where you're actually able to link very important cellular circuits to metabolites that are taken in from the outside, on top of all the regulating growth factors and in cell-cell interaction that you will have. So some time ago, I was looking for Professor Harald Zittich. I didn't find him because he's always so busy that he's going away. But our common boss, Misha Freismuth, is here. Some time ago, we decided this was too big a task to do by ourselves. And we, with some luck and some help, we wrote a piece for the journal Cell that said, this is too big a job for us. Many people should study this kind of transporters. And following that, the European Union issued a call, together with sort of FPIA, a call for a joint effort between pharmaceutical companies and the public to sponsor basic research on transporters. And this was called Resolute. We won it and we coordinated. And it was a 5-year effort. And then we had just here in Vienna, in Palais Niederösterreich in September, the final wrap-up conference. Unfortunately, it's not really a wrap-up because we still have a lot of work to do and write up the papers, but it was an incredible community. So what I'm telling you now is not the work of my laboratory alone, it's the work of literally 100 people. Okay, so how to do this? How to try to understand the activity of these transporters? We generated a lot of 1,500 almost cell line models. So cell lines where a gene is inactivated, and then first you inactivate the endogenous gene, and then you create a system where you have an inducible cDNA that is able to reinstall. And we did it in different kind of cell lines. And this is, I'm not going to go you through all of this. We also did some cell lines that where the endogenous gene was not expressed because it was impossible to find the proper cell line. And so once you have this tool, you have a cell that is either expressing or not expressing a transporter, you can try to ask a lot of questions. For example, what are the changes in metabolites? What are the changes in transcripts? And so on and so forth. And this is something that we did in order to find out what is this transporter doing. And we have almost 500 to work on. And this is a busy slide that gives you a summary of what we have done. Imagine this to be each a section of the knowledge that we are trying to create. The first one being the analysis of all the transcript, the second slice being the analysis of all the metabolites, then the third being the interaction of the proteins that are interacting with your sort of transporter of choice, then genetic interaction, where you inactivate one and what happens to inactivation of the other ones, transport assay, which are of course very important, structural similarity, I showed you this tree, but also genetic variation. There are a lot of interesting genetic variations in our genomes that are dealing with transporters. And all of this coming together, we hope to be able to then, not only based on the evidence that we create in one dimension, but by making connection between the different dimensions to come to some sort of hypothesis as to almost all of these transporters. And the metaphor is a torchlight that is built by these batteries of different elements and trying to understand this. Now, this integration of data is something that is keeping us very busy. It actually works. There are several ways how you can integrate multidimensional data that is sort of object specific, and there are several solutions that it's beyond my ability to explain, but we're working hard on it. And certainly, integration of all this data is what we call our knowledge domain, and all of this is actually accessible at a portal that is open access. You can, if you're interested in transporters, you can have that site, and it will navigate you. It will give you the data, it will give you the structure, it will give you information on individual transporters. Now, very briefly about drug targets, because this is a group of people sitting in this audience that are actually interested in doing something. There is an industrial application, there is a biomedical application, there is an environmental application. And so transporters and drug targets are really, it's an interesting story because some of them are very well characterized and some of them are very poorly characterized. But the majority of transporters do not have chemical probes. And I mentioned already some of the very famous ones, in fact, monoamine transporters. And right now, there's only about, out of these 450, only some 30 that are properly targeted by chemical agents, although the number is growing every day. And there is a lot of, the challenge is that there is a lot of different chemical matter that binds to these transporters, because some of them transport peptides, some of them transport ions, so they differ a lot in what they can. And clearly, we are not in a situation where we actually know what they are, how to drug them, and many of them we suspect have additional binding sites. This is just a cartoon that shows that the distribution of assays over the different transporters is very asymmetric. Some of them we have a lot of assays, and some of them we have 0 assays. So this is something that needs to be tackled, and I'm not going to go into detail. But based on the genetic interaction map that we had done in the lab, a student, Wojtek Dvorak, thought that maybe what we can do is can use this genetic redundancy, the fact that often, I didn't tell you this, but there are 12 or more glucose transporters in our genome. If you knock out one, you're not able to block the entire ability of a cell to take up glucose. And there are many, many amino acid transporters and so on and so forth. So what you do is you try to understand a situation where the function of an SLC, when inactivated, is not sufficient to kill a cell. The cell survives in the normal conditions that we have in tissue culture, which are totally artificial. In those conditions, the cell can grow. If you inactivate the other gene, the cell can grow. If you inactivate both, the principle of synthetic lethality, the cell cannot grow. So if you construct your cellular screening assay for drugs so that the cell is depending on only one of the transporters, and you screen all the other conditions, such as the wild type, or the one that has inactivated the other transporters as control, you should be able to identify drugs that only are working because of the presence of that particular. And that is exactly what we've done with 2 sort of mercarboxylate transporters, actually 2 lactate transporters, SLC16A1 and A3, MCT1 and MCT4. And this turned out to work very well. So if you take 90,000 drugs that is composed of Stefan Kubitschek's library in our institution, and you make a screen against a gene, a cell that lacks the gene SLC16A1, then the cell depends on 16A3, which is the one that you target. And so you get your 1,400 compounds, and then you screen them for the other 2 cell lines, which is wild type, and then it should not kill, or the 16A3 knockout, it should not kill because it is not targeting 16A1. This is very, very simple. It's as simple as it gets in drug discovery. But it worked like magic, so we identify a series of compounds that were, from the very beginning, entirely specific for this particular transporter that we were, without knowing, we should not have known anything about the transporters except that it was essential for it. And this is a lot of work that I'm summarizing here, where almost any genetic combination is not killed by the drug, this new one, but only if you lack 16A1 because you're depending on 16A3, proving this. And then in the meantime, it was feasible and possible to derive models, because we don't have the structure of this to model it and to understand exactly where the specificity comes from, because then you have all these different paralogs, because we also tested 2 different paralogs, and you can decide the drug is specific for only one, because it has this amino acid in the binding pocket and not these two others. Even if we don't have a real structure with the drug, we think that it is probably true because of the ability to swap and recreate sensitivity. And this is published, so I'm not going to go into much detail. Another case, and this is the last of this case, is there was a very interesting SLC called SLC15F4 that is a protein involved in innate immunity. It helps recognize something that is not entirely clear in the lysosome, and it assists toll-like receptors 7 and 9 that are nucleic acid binding innate immunity receptors that are pro-inflammatory. They actually normally are just calling for action and say there is a pathogen. If you have a bacterium invading, if you have a virus, then the nucleic acid that is not modified is recognized by the endogenous system of toll-like receptors, and it does. Now, the signaling to IRF5, which is a very important transcription factor for innate immunity, is dependent on this SLC15F4, and we had previously identified an adapter called TASL that is actually binding to 15F4 and is essential for this function. Without TASL, the whole pathway stops being. And we're talking about an important pathway because each of all members is actually associated with an autoimmune disease called lupus erythematosus. So SLE, systemic lupus erythematosus, you have mutation in toll-like receptor 9, in 15F4, in TASL. All of them are associated with this disease. And so what happened here is we took another notion that we knew about the protein, and that is that if you don't have the interaction between 15F4 and TASL, TASL gets degraded. So it's a simple assay that we derived. We say we are looking for drugs that when they hit the cell, they degrade TASL. And you can try to do that in a specific way because you can also have an endogenous other protein, in this case it will be red fluorescent instead of being green fluorescent, that should not be affected by the drug. And so Andras Borshemeni, that did the screen together again with the Kubitschek lab, was able to identify a molecule that we call fiblin because the mouse mutant of 15F4 is called Fibl. It has a phenotype that's associated with poor immune system. And fiblin was reproducing this situation. And so what fiblin does, it is bound from the lysosome internally to the SLC. The SLC assumes a structure that is incompatible with binding to TASL, and TASL gets degraded. Maybe this now works. It's a bit of a slow movie, but let me see. It gives you a little bit of a sense of the situation. This is fiblin binding. We have the outward open situation where the drug can bind. Else, when the drug is not binding, it is open to the cytosol, the SLC, and there it is where TASL can bind. It should come in a minute. This is just the tail of TASL is engaging. So it's a beautiful allosteric mechanism that leads to degradation of TASL, and this is something that now a biotech company that we started is taking on very seriously. So very briefly, these are the two heroes of the story. We think that solute carriers will become as important drug targets, and we will hear more about this, as are GPCRs or kinases or proteases or channels in future. And I finished by saying, some time ago, more than 10 years ago, we started a project called Genome Austria that was not supported by the system. We were ahead of our time, and I had my genome completely sequenced. Not too many interesting things came out. It's actually public. You can look at it. I had for many years, I had at the bottom of my email, this is my genome. If you find anything interesting, tell me. Just to tell you, because this is a country that is so scared by genes, and all my genes are online and nothing happened. Nobody fell in love with me because of this. Nobody stopped my friendship because of this. Nothing happened. So the genes are not that interesting if you don't know how to read it. But the point is that the most interesting thing was that I had 3% Neanderthal, as you can see from my nose, without sequencing the genome. There are a million such slides, and these slides contain a typical example of what you call common polymorphisms in blue. It's a bit difficult to see, but there are some mutations in blue that are very probably common to all of us in this room, depending on our ethnic origin. But then there are some that are quite private. And these are the ones in red. And I have many mutations, actually 8 mutations in SLC genes, one of which, for example, is a heterozygote mutation in SLC34A1, which is a phosphate transporter. And I believe, I'm convinced that some of the kidney stones that I have and my brother has actually come from mutation in this gene. And I tell you this because as you sit here, you have many mutations in your transportome that will allow you to be interacting differentially, chemically with the environment in ways that your generation will certainly find out. And I like this slide because it shows that each one of us during a lifetime takes up tons of food, a lot of things that we drink. Some of this stuff goes out of our body again, obviously, in form of water, in form of urine, and so on and so forth. And we are like a filter. We are like taking up all of this, and much of this actually does go through transporters and actually does go through membranes. So in the course of our lifetime, and this is important because if you take on, and there are some beautiful posters that speak about this, if you think about the microbiome that is metabolizing a lot of different sort of nutrients and so on and so forth, you understand that there is an insertion of the human body as a filter of what is the environment. And you could thus imagine, because we know this, that if you're an animal, you're a plant, you're a fungus, you're a bacterium, you're likely to use the same blueprint of transporters because they're evolutionarily the same. Most of our transporters are like that. So that you say the ecosystem is a big traffic of chemical matter through transporters of the different components of the ecosystem. And I say this because I think that in future we need to understand this chemical integration of humans with the environment in order to be able to make an environment that is healthier for us and that eventually we're able to improve. So there are many open questions, and because I'm not going to go through all of which, except that we are clearly interested in understanding what I just described to you, and these are two students wearing transporter costumes because you only understand transporters if you are. So I finish with a little bit on the title of my talk. It has to do with social engineering. So imagine the ability of making institutes, or the ability of organizing departments, or the ability of running a foundation as part of the scientific tool. You think that all you need to do is to have your pipettes, to have the money to the reagents, and a roof over your top in your laboratory. But it takes much, much more. There is an educational system, there is a social system, there is a system that allows you to hear good lectures, there is a system that allows you to have good colleagues, and there is a system that recognizes your work. And so we have been, for example, advocating that there are research institutions, such as the one that I work for, the CeMM, that are actually very important to complement the activity of universities, because universities having so much training duty and so much also research duty connected to the teaching duty, they don't have a lot of agility in checking out new stuff. And we wrote a piece, all the directors of 15 different institutes wrote a piece that summarizes this. So the modern research centers can foster a cooperation, innovation, and sort of better than a lot of other settings. And I mention this because without taking up responsibility, and there are some people in the audience who hear me, without being able to take up responsibility for some of the organization, and some of you will become rectors of universities or head of institutes in the audience, you will not be able to do research the way that it should be done, which is very cooperative and is very, let's say, open for innovation. Science is our medicine is our slogan. I'm not going to tell you about the center that I work on because I think I have already exhausted my time. We belong to the Academy of Sciences. The Academy of Sciences, as you know, is a very important non-profit academic organization that is complementary to the university system since 175 years in this country. And we have a few important principles. One is that most of the people are young. One is that everybody is kind and very collaborative, and we praise excellence. And I just show you, these are the different investigators. Many of them are outside our building, but are connected to us through an adjunct system. Many of them, actually 12 out of 19, have an ERC grant or have had an ERC grant, an important one for proof of the excellence. And they are very, very collaborative. And this is the notion that I want to leave you with. And that is that we measure this because we have many other institutions that we compare ourselves, and this institution is 4 to 5 times more collaborative in terms of numbers of papers that individual PIs publish together with other PIs, and also the number of PIs that each PI works with. And that is, I think, a very significant number. I'm not implying this is the only way, but I'm implying that this is a very nice and efficient way to maximize synergies and to create the kind of innovation culture that I think has allowed me to do what I'm doing. All of this on the shoulder of giants. The mentors depicted here have all been testimonials and supporters of our center. All of them, the one depicted here, are Exner medalists. I'd like to point out Uwe Sleiter because he's in the audience. I would like to thank him in particular. He's also an artist. You can't quite see it, but this is some of the art pieces hanging behind him. And I know that I'm an old man and I sound a bit boring, but you can only do this because you have people that are supporting you, who have paved the way, who know and who sat in committees, have done sort of what appears like a boring part, which is to make optimal conditions for you to be able to research. So I would like to thank them very much. And very briefly, it's because of them that I'm now taking on a new challenge. I don't know whether some of you knows the White Lotus. I know that Georg Winter knows these are the different sites of the White Lotus. It's a Netflix series, and this is Sicily in southern Italy, in the center of the Mediterranean. And close to Palermo, we're making a new center. And this new center that you see here, these are 2 chromosomes, they're built, is what I call the European Crick, because it is actually done by the same architect that have done the Francis Crick Institute in London, but it's Crick in the sun. It will open in 2 years from now, and it will house some 600 people. And very briefly, a little overview on where it is. This is the plain of Carini, which is 20 kilometers from Palermo next to the airport. And this is the center. These are the 2 chromosomes that you see. And these are all the additional buildings that are like a guest house here, an auditorium here, and auditorium here, administration, and a few other buildings that are coming up. So if you're interested, and many of you are working on the interface with environment, this is what we're going to be doing. We're going to be working at the interface between the human system and the environment in all its aspects, including aspects that affect the central nervous system, 600 people, close to the seaside, funded by a foundation. What is very interesting is that everything associated with the Institute will try to be resembling the kind of things that we want to study. And so if you're interested, apply now. I'm keen to do that before advertising because I want to keep it as international as possible. I would like to thank a lot of people. Some of them were mentioned during the talk. It's difficult to, these are literally hundreds of people. And yeah, thank you very much for your attention.